So next is the M phase or the mitosis phase. Mitosis phase or mitosis division is a type of division which is called equational division. Because in this type of division, the chromosome number is restored in the daughter cell. That is, if the parent cell is diploid, the daughter cells will also be diploid in number. If the parent cell is haploid, the daughter cells also will be haploid in number. But generally mitosis takes place in diploid cells, except for some few lower organisms. M phase starts with karyokinesis and ends in cytokinesis. So mitosis phase starts with karyokinesis. Karyokinesis means division of the nucleus, and it generally ends in cytokinesis, where the cytoplasm divides and finally two new daughter cells are formed. So as it starts with karyokinesis involves 4 stages. What are the 4 stages of karyokinesis? First is the prophase, second is metaphase, anaphase and telophase. The first phase is prophase. Prophase, in this phase, the chromatin condenses to form chromosomes. Chromatins are generally thread-like structures which are found in normal cell before the cell division occurs. But when a cell is ready to divide, it has to undergo various changes. The first change is that a chromatin will change into chromosomes. That is, suppose this is chromatin. Chromatin are thread-like structures. Chromatin will change into chromosomes, which are condensed structures. These will change into condensed structures. These are called as the chromosomes. Second changes is chromosomes appear as sister chromatids which are joined by the centromere. Suppose this is one chromosome, this is other chromosome. So these two chromosomes will join together as sister chromatids. By the help of, this is the centromere. And the two chromosomes are now called the sister chromatids. Spindle fibers are formed in a cell that is ready for division, that undergoes prophase. In that phase, the spindle fibers are formed. Centrioles migrate towards the opposite poles and the nuclear membrane disappears. So this we will understand with an example of a diagram. Suppose this is a cell that is now undergoing prophase. This is the nuclear membrane which is disintegrating. This is the nuclear membrane. After that, this is the centrioles which have divided in the cytoplasm, and this is early prophase, we can say. Early prophase. This is the early prophase. Here the chromosomes are now condensed and sister chromatids are formed, and centromeres are present between the two sister chromatids. This is this structure we can see here. This is one, this is other, and this is the centromere. During the late prophase, what will happen? This cell will now, the nuclear membrane will disintegrate and the spindle fiber will move towards the opposite poles, carrying the centriole, and the chromosomes will be present, arranged in this manner. The nuclear membrane has now disintegrated and centrioles or spindle fibers move towards the opposite poles. Chromosomes now condense, come together with each other in the form of the sister chromatids, and there is a presence of centromere. And this is all about the prophase. This is early prophase, we can say. This is the late prophase. This is the late prophase, and this is the early prophase. So the next phase is the metaphase. Metaphase, during this phase, the morphology of the chromosome is clearly studied because the chromosomes are now in a very condensed state and they are clearly visible under the microscope. So at this position, we can clearly study the structure of chromosomes. Now the chromosomes in this stage align at the center of the chromosome. At the center of the chromosomes, now you can see that the chromosomes align themselves at the center of the chromosome or the equatorial plane of the chromosomes. The plane of alignment of the chromosome is called as the metaphase plate because they are aligned in a straight line at the equator or the center of the cell. So this phase can be easily identified during under the microscope, as the chromosomes are arranged during the equatorial plate. And the next phase is the